I am a Consultation-Liaison psychiatrist and academic physician. Welcome to Polypharmacy MD. Today, we conduct a clinical deep dive into the molecule that promised to solve the greatest paradox in psychopharmacology, Risperidone. In the early 1990s, clinical psychiatry reached a definitive turning point. For nearly 4 decades, the field was characterized by the era of typical antipsychotics. We relied on high-potency dopamine D2 antagonists, such as haloperidol and the phenothiazines. While these medications successfully addressed the positive symptoms of psychosis, they often resulted in significant iatrogenic motor impairment. On the hospital wards, this was colloquially described as the Thorazine shuffle. It was a visible constellation of Parkinsonian tremors, a mask-like facies, and the persistent risk of tardive dyskinesia. At that time, managing schizophrenia often involved inducing a state of iatrogenic parkinsonism. To illustrate this burden, consider a representative clinical encounter. A 45-year-old male with chronic schizophrenia is admitted to a medical unit. He has been stabilized on a high-dose regimen of haloperidol for several years. While his auditory hallucinations are absent, his physical state is marked by significant extrapyramidal side effects. He exhibits marked bradykinesia and a rhythmic resting tremor in his hands. His facial expression is fixed and lacks emotional reactivity. When he attempts to walk, his gait is short and shuffling. In this clinical scenario, we have suppressed the psychotic symptoms, but we have done so at the cost of the patient's motor coordination. The medical community required a successor to clozapine. We needed a molecule that could offer atypical efficacy, addressing the symptoms without significant neurological adverse effects, but without the hematologic risk of a granulocytosis that mandated strict and costly monitoring. We begin with the introduction of Risperidone, known generically as Risperidone and launched in 1993 by Janssen Pharmaceuticals. It did not merely change the management of schizophrenia, it fundamentally re-engineered the pharmaceutical marketing landscape. It was the molecule that promised to be the Goldilocks of psychopharmacology, potent enough to suppress a psychotic episode, but theoretically sufficiently tolerated to avoid the extrapyramidal symptoms of the past. However, the history of Risperidone is not solely one of chemical innovation. It is a cautionary analysis of how a revolutionary molecule can be influenced by corporate strategy. This is the narrative of the rise of the Atypicals, a period where medical science intersected with aggressive commercialization. We will explore the neurobiology of serotonin-dopamine antagonism and the historical use of the LSD model of psychopathology. We will examine the long-acting formulations that altered outpatient adherence, and we will review the marketing tactics that led to one of the largest Department of Justice settlements in medical history. From the sales strategies designed to influence prescribing patterns to the clinical data that revealed a significant association between this agent and the development of breast tissue in pediatric patients, we are covering the full technical and historical scope. This is the complete history, marketing, and mechanism of Risperidone. The development of Risperidone began in the laboratories of Janssen Pharmaceutica in Beerse, Belgium. In the late 1980s, the laboratory, under the direction of Paul Janssen, was operating at a high level of chemical innovation. Their primary objective was to solve the central paradox in psychopharmacology, achieving the superior clinical efficacy of clozapine without the risk of a granulocytosis. To find a solution, researchers moved away from traditional butyrophenone and phenothiazine structures. They shifted their focus to a novel class of compounds known as benzoxazole derivatives. While early research in the Janssen labs had utilized hallucinogen models to study reality distortion, the focus narrowed to the precise engineering of receptor affinities. The goal was to synthesize a molecule that could target the brain with higher selectivity than the non-selective dopamine antagonists of the previous generation. To understand why this medication became both a clinical staple and a legal liability, we must examine its specific pharmacodynamics. Risperidone's behavior in the human brain is highly dose-dependent. In clinical practice, I often see the consequences of this dose-response curve. Once the daily dose crosses the 6 mg threshold, the protective effect of the serotonin 5-HT2A blockade is effectively overwhelmed. At this point, D2 occupancy in the striatum reaches the critical 80% mark, and the agent begins to mirror the side effect profile of high-potency typical antipsychotics. The most distinct mechanism of risperidone is its effect on the tuberoinfundibular pathway. Under physiological conditions, dopamine exerts tonic inhibition on the release of prolactin from the anterior pituitary. Because risperidone crosses the blood-brain barrier with high efficiency but also exhibits high affinity for receptors in the pituitary. It antagonizes this tonic dopaminergic inhibition. This results in significant dose-dependent hyperprolactinemia. There are specific pharmacological reasons why risperidone impacts the tuberoinfundibular pathway more significantly than agents like haloperidol or clozapine. While haloperidol is a potent D2 antagonist, risperidone often produces higher prolactin levels due to its unique pharmacokinetics and anatomical distribution. First, consider the role of the blood-brain barrier. The anterior pituitary gland is located outside the blood-brain barrier, meaning its D2 receptors are exposed to peripheral plasma concentrations of the drug. Risperidone and its active metabolite, 9-hydroxyrisperidone, are substrates for the P-glycoprotein efflux transporter. This transporter actively removes the molecule from the central nervous system, but because the pituitary lacks a traditional blood-brain barrier, it is not subject to this P-glycoprotein-mediated efflux. Consequently, the concentration of risperidone in the pituitary remains significantly higher than in the striatum. Positron emission tomography studies have demonstrated that while striatal D2 occupancy may be in the 60 to 70% range, pituitary D2 occupancy often approaches 100%. Second, the binding kinetics of risperidone contribute to this sustained effect. The blockade at the lactotroph D2 receptors is a function of the dissociation constant, or K-off rate. Unlike atypical antipsychotics such as clozapine, which exhibit fast-off kinetics, dissociating from the D2 receptor within seconds, risperidone possesses a significantly slower dissociation rate. This leads to a persistent occupancy of the receptor, which prevents the intermittent dopaminergic signaling required to suppress prolactin secretion. While clozapine allows for transient receptor availability, risperidone provides a near-continuous disinhibition of the lactotrophs. This is why risperidone is frequently identified in clinical literature such as the American Journal of Psychiatry as causing prolactin elevations that often exceed those of both typical and other atypical antipsychotics. Clinical observations documented in publications such as the American Journal of Psychiatry indicate that risperidone induces prolactin elevations significantly beyond those observed with other atypical antipsychotics. In female patients, this frequently leads to amenorrhea and galactorrhea. However, the most significant clinical sequelae occur in the pediatric population. In young boys, sustained hyperprolactinemia can result in gynecomastia, which is the permanent development of female breast tissue. This association between the agent and pediatric endocrine disruption was a pivotal discovery that shifted the clinical narrative from innovation to cautious management. As the clinical landscape evolved, so did the delivery systems for the molecule. The development of the long-acting injectable, or LAI, was a pivotal moment in psychiatric history. Risperidone Consta was the first atypical medication to utilize Metasorb microsphere technology. From a clinical perspective, this was a highly effective tool for managing patients with chronic schizophrenia who exhibited poor medication adherence. It transitioned the treatment from a daily oral regimen to a biweekly injection, theoretically reducing the frequency of iatrogenic hospitalizations. However, this clinical innovation also served a corporate purpose. By creating new patented delivery systems like the MTAB and the long-acting injection, the manufacturer was able to maintain market dominance and premium pricing even as the original molecule approached its patent expiration. Finally, the company successfully isolated the active metabolite of risperidone, known as 9-hydroxyrisperidone. This was marketed as a separate drug, paliperidone, by utilizing an advanced osmotic release oral system known as OROS. They were able to offer a once-daily dosing profile that further extended the life cycle of the risperidone lineage. To provide the necessary regulatory context, we must establish the clinical baseline of the authorized applications for this agent. The FDA-approved indications for risperidone include the treatment of schizophrenia in adults and pediatric patients aged 13 to 17 years. It is also indicated as monotherapy or as an adjunct to lithium or valproate for the treatment of acute manic or mixed episodes associated with bipolar I disorder in adults and pediatric patients aged 10 to 17 years. Furthermore, it is approved for the treatment of irritability associated with autistic disorder in pediatric patients aged 5 to 16 years. Establishing these specific parameters is critical when analyzing the subsequent transition of the drug into populations and conditions where regulatory clearance was absent. The commercial success of Risperidone was not merely the result of this approved neurobiological profile, it was the product of a sophisticated and often aggressive marketing strategy designed to position the drug as a universal solution for agitation and behavioral disruption. As a consultation liaison psychiatrist and academic physician, I have observed how pharmaceutical positioning can influence clinical decision-making on a significant scale. One of the primary strategies was the haloperidol comparison. In many of the early clinical trials conducted by Janssen, risperidone was compared to exceptionally high doses of haloperidol, often as high as 20 milligrams per day. By comparing their new agent to an overdosed typical antipsychotic, Janssen was able to claim that risperidone had a superior safety profile regarding extrapyramidal side effects. This established a powerful narrative for sales representatives, even though lower, clinical doses of haloperidol might have produced a similar adverse effect profile. In the late 1990s and early 2000s, the marketing reached beyond the clinical office and into the general population. Magazine advertisements and publications such as Time and Newsweek featured serene imagery, often of elderly individuals or calm family settings, accompanied by text emphasizing a new era of psychiatric stability. These advertisements were carefully crafted to associate the drug with safety and modern scientific progress, contrasting it with the perceived harshness of older medications. On television, the messaging was even broader. While many advertisements focused on the approved indication of schizophrenia, the subtext often suggested broader applications for mood and behavioral control. This subtle positioning established the framework for the most controversial chapter of the risperidone story, the promotion of off-label use. To maximize market share, Janssen allegedly targeted populations for which the agent lacked FDA approval. According to official reports from the United States Department of Justice, sales teams were incentivized to promote the drug for agitation in elderly patients with dementia. This was a patient population where the drug eventually earned a boxed warning due to an increased risk of cerebrovascular events and mortality. Furthermore, the company was accused of paying kickbacks to Omnicare, the largest pharmacy provider for long-term care facilities, to ensure that risperidone was the preferred treatment for elderly residents. This aggressive expansion of the drug's clinical footprint eventually triggered a massive federal investigation. In a strategy known as the Popcorn Program, sales representatives reportedly distributed materials encouraging the use of risperidone in pediatric patients for ADHD and autism, long before the drug was cleared for use in children. This push into the pediatric market led to one of the most significant clinical complications of the era, pharmaceutical-induced gynecomastia. Consider a representative clinical scenario. A 12-year-old male with irritable behavior associated with autistic disorder is initiated on risperidone. While behavioral agitation improves, he presents 8 months later with palpable bilateral subareolar masses. This is not pseudo-gynecomastia or simple adipose tissue accumulation. It is a proliferation of glandular breast tissue. The sustained hyperprolactinemia has triggered ductal hyperplasia and a secondary suppression of the hypothalamic pituitary gonadal axis. For an adolescent male, this physical transformation often reaches permanence that necessitates surgical intervention through bilateral mastectomies. The corporate history of this complication is highly documented. In 2013, the Department of Justice announced that Johnson & Johnson and its subsidiary Janssen would pay more than $2.2 billion to resolve criminal and civil liability. The litigation brought to light internal documents suggesting that the company had withheld data regarding significant prolactin increases in children as early as 1993. Furthermore, a 2003 study published in the Journal of Clinical Psychiatry was scrutinized after unsealed court exhibits suggested that Janssen employees had internally identified data regarding the persistence of hyperprolactinemia, data that was allegedly omitted from the final published manuscript. This intersection of corporate strategy and clinical risk remains a defining moment in modern medical history. Despite these controversies, risperidone remains a cornerstone of modern psychopharmacology. Its legacy is a complex tapestry of revolutionary neurobiology and institutional caution. As a consultation liaison psychiatrist and academic physician, I view the history of Risperidone as the moment when psychiatry officially entered the era of the SDA class. The success of this molecule paved the clinical pathway for subsequent atypicals like olanzapine, quetiapine, and ziprasidone. Each of these followed the molecular blueprint established in the Janssen labs, targeting the serotonin 5-HT2A receptor as a means to mitigate the extrapyramidal side effects of D2 blockade. Perhaps the most significant clinical legacy is the development of its active metabolite, 9-hydroxyrisperidone. Marketed as paliperidone, this compound represents the refinement of the original risperidone structure. By utilizing the OROS delivery system, clinicians are now able to provide more stable plasma concentrations, reducing the peak-to-trough fluctuations that were often associated with the adverse effect profile of the original tablet. Furthermore, the innovation of the long-acting injectable has continued to evolve. From the biweekly injections of Risperdal Consta, we have transitioned to formulations that allow for monthly, 3-month, and even 6-month dosing intervals. These advancements have drastically improved the management of chronic schizophrenia, reducing the risk of relapse and iatrogenic hospitalization. The history of risperidone is a dual narrative. It is a story of how a significant molecular discovery provided a necessary alternative to the high-potency typical antipsychotics of the past. Yet it is also a reminder of the inherent risks when aggressive corporate strategy overlaps with clinical practice. Today, risperidone is available as a low-cost generic medication used daily by millions. It serves as a fundamental tool in the psychiatric toolkit, provided it is used with the clinical nuance that its dose-response curve requires. The Goldilocks molecule of the 1990s may not have been as perfect as the early advertisements suggested, but it forced the field of psychiatry to evolve, demanding greater transparency in clinical trials and a more profound understanding of pediatric neurobiology. For Polypharmacy MD, I am a consultation-liaison psychiatrist and academic physician. If you value this high-level analysis of medical history and neurochemistry, subscribe for our next deep dive into the molecules that shaped the modern world.